Our next speaker is Michael McMurray from the University of Colorado.
Great, thanks for coming this morning. So when I learned about the yeast life cycle, originally I think it was from what I now consider to be sort of the yeast geneticist perspective, which is exemplified, I think, from this figure from an Ira Herskowitz review that shows how diploid cells, when starved for nutrients, can undergo meiosis and produce 4 haploid spores that are encased within the so-called ascus, the old cell wall of the original diploid mother cell. So Ascus was actually a giant from Greek mythology, and Zeus slayed him and flayed him and made a bag out of his skin, which is appropriate because you learn from this figure that that Ascus wall must be destroyed in some way to release the 4 spores and allow them, when exposed to nutrients, to begin the process of germination and resume mitotic division. So when I was a postdoc in the Thorner lab, one time Jeremy told me that if you put 2 cells of opposite mating type together on a plate, 100% of the time they will mate immediately, which suggests to me that you, in order to get these spores of opposite mating type to bud, you have to disperse them from the ascus. So I was surprised when I was studying, at the time, the persistence of septin proteins through sporulation and germination. I was lazy and just skipped the spore purification step and left intact asci together and just re-exposed them to nutrients. And I was really surprised to find that in many cases those spores could bud immediately despite the presence of the ascus wall, and they didn't in fact mate. And so that said that breakdown of the ascus wall is not in fact a prerequisite for germination and budding. And also it led me to the startling conclusion that these sister spores, these meiotic sister spores, actually have a choice about whether or not to mate with their other sisters. And so I had sort of forgotten about these results for a few years until my grad student Lydia told me about some experiments she'd done in which she was watching germination from asci that were either intact or in which she'd removed the ascus wall by exposure to zymolyase. And she noticed that plenty of cells did bud when the ascus wall was intact, but of course lots of them also mated within the ascus. But if she removed the ascus wall, she saw a big increase in the amount of cells that budded as their first decision instead of mating, which suggested that the persistence of the ascus wall seemed to disfavor mating and promote budding. And this made us try to go back and try to look in the literature for evidence of what was the fate of the ascus wall upon germination if you don't remove it otherwise. And the literature is really sort of a mixed bag, pun intended. You can see these more recent studies that use beautiful scanning EMs to look at germination, but unfortunately, they digested away the ascus wall prior to germination, so it doesn't really help address the issue. They did point out these interspore bridges that connect the 4 spores together, even when the ascus wall is degraded, and that'll become important later in the talk. But in order to find out more about this, I had to look back in the 70s to some transmission EMs, this time which showed that when spores are swelling or budding, the new cell wall that's produced is able to erupt from underneath the mother wall or ascus wall here, and thus break it locally, but without causing its global disintegration. So this really suggested that ascus wall breakdown could be a very local event. And now, it wasn't clear from those old EMs whether the original mother cell's plasma membrane was persisting through this process, and so Lydia did some FM464 labeling of cells during sporulation and saw that early on the mother cell's plasma membrane seemed to protect the spore membranes from labeling with FM464. But then as sporulation progressed, the structural integrity of the mother cell's plasma membrane started to break down, and you could start to get some labeling of the spores. And then late in sporulation, there appeared to be no plasma membrane left, and only the spore membranes would label. So we think that during germination, all we're dealing with is an old cell wall here. So then Lydia did a more direct experiment of sort of a calcofluor pulse chase to look at the fate of the ascus wall, and the results were actually quite striking. You can see here, upon germination, buds emerging from holes in the ascus wall, and even some spores appearing to be able to pop out of these holes. But the ascus wall otherwise is able to stretch elastically and still hold on to some of the spores, despite the fact that some of the cells are being able to exit the ascus. So this was quite unexpected for us. And around the same time, we had seen a talk at a meeting from Ali McClure, who was then a student in Danny Lev's lab, and she had also become interested in this decision about whether to bud or to mate upon germination, and she wanted to know if it was under any sort of genetic control, and so she obtained a collection of genetically diverse strains and used time-lapse microscopy to watch them and see whether their first decision was to bud or to mate. And these movies aren't working here, but she was, with a trained eye, able to look at all the 4 spores and decide how many of them would bud first versus mate. And she saw a nice variety of these decisions among these different strains and could conclude that there could be some genetic differences underlying these variation. So as a yeast geneticist, I wanted to try to transform the cellular event into a colony growth phenotype. And so I developed a strain that I call the German, which has G418 resistance under a haploid-specific promoter. And so if the 2 compatible partners within a tetrad mate immediately, you do not get any growth of a colony on a G418 plate. And so I then separated 40 tetrads in which the ascus wall was either intact or degraded with zymolyase. And you can see here that if these mating events are randomly assorted, that the probability of mating is about 40%, but if you degrade the ascus wall, that drops to below 30%. So the ascus wall, again, does seem to promote mating and disfavor budding. And satisfyingly, these numbers were actually quite consistent with Ali's observations from microscopy, because she had included 2 lab strains, and those also showed about 40% mating as the first decision. And so this makes us wonder if some of the differences between these different strains could be due to differences in the properties of the ascus wall during germination. And now, I recently sort of discovered that this observations about ascus persistence during germination had actually been documented almost a century ago here. And in this book chapter, they noted that the ascus wall becomes thinner during germination and finally breaks, but that it acts as a plated veil, which is able to persist and retain the ascospores. And more about the plated veil in a second. But they also said that most of the buds appear on the surface, but occasionally there are some buds that may appear on the interior of the ascus, which really surprised me. How could buds get trapped within the ascus? So first, here's a picture of a plated veil that I took from Pinterest, and I think is actually quite appropriate. And second, when I was thinking about the buds getting trapped within the ascus, I realized I'd actually seen this before in my own published data, which I've actually shown you a few slides ago. And that was when I happened to see some dyads, so asci in which only 2 spores are produced, which happens pretty frequently in strains of the S288C background. And you can see here that one of these buds was able to easily penetrate the ascus, but one of them seemed to be trapped within the ascus, which you can see with the fluorescent septum tag that I have here, the bud neck there. And so I looked at some more dyads using septins to mark the bud neck, and saw plenty of occasions in which indeed the new bud would get trapped within the ascus. Whereas it was basically impossible to find this happening with triads or tetrads, which suggested to me that there must be some threshold of some sort of activity that's required in order to thin the ascus to make it thin enough that a bud can easily penetrate it. And that if you have only 2 viable spores within the ascus, you sometimes don't reach that critical threshold. And I suspect that probably secreted cell wall hydrolases represent the relevant activity, because that's what Pombe cells use to very efficiently destroy their ascus wall during germination, long before budding ever begins. So in the last few slides, I'd like to provide some bigger picture context for when this inter-ascus mating or bud versus mate decision can become really important, and some opportunities for when ascus wall breakdown might sort of normally occur. So wild Saccharomyces are largely diploid and spend most of their time proliferating mitotically, which gives them many opportunities to accumulate sequence variations or mutations, usually in a heterozygous state, and only rare opportunities to undergo meiosis and expose any new recessive alleles to selection. And upon meiosis, then, the decision upon germination of whether to mate or to bud has huge consequences for how much of that variation is exposed and how much is eliminated. And this is especially true when you consider homothallic strains that are able to switch mating type and mate with their own progeny. And Michael Kanopis thought very deeply about this process and has realized that the tight centromere linkage of the mating type locus assures that centromeric loci tend to remain heterozygous in diploids that are produced by intra-ascus mating, and that because of this, evolution may have driven what you see in the Saccharomyces genomes, which is that essential genes are clustered and tend to be biased towards pericentromeric locations. Now, people have also recently realized that most wild cells strains spend at least some time, perhaps, in the guts of insects, and that the insect gut has enzymes that can break down the ascus wall. And this has been proposed by others to perhaps promote outcrossing or inter-ascus mating between asci. But I think our results suggested it could also promote budding and therefore loss of heterozygosity. So Michael Kanopin noted that this decision about budding versus mating and inter-ascus mating is really important for sequence variation alleles that do not allow mitotic proliferation, but do allow germination and mating, because such alleles could actually be rescued by the act of inter-ascus mating and therefore persist in the population. And so I wanted to be able to develop a strain to test this, and so I developed this one that I call the Swiss, in which I have a deletion of the TEM1 gene, which is required for exit of mitosis, marked with G418 resistance. And so the only way to get a G418 resistant colony is if you have immediate mating by two sister spores before they attempt to bud. And you can see here that digesting the ascus wall, again, seems to disfavor mating and promote budding. And so the final scenario I want to talk to you about, in which I think this inter-ascus mating is extremely important, is in de novo speciation. And so the strains of the species of the Sensu strictu collection are able to form viable diploids that can sporulate, but the vast majority of the spores are dead because those sequence differences prevent, as Neil just mentioned, proper chromosome segregation during meiosis 1. And so there's very few of the viable spores are alive because for any of the 16 chromosomes, their mini-spores fail to inherit even a single copy. But those rare viable progeny do have unique properties and are potentially new species. And viable progeny, I noticed though in these studies, have always been isolated from isolated spores in which there was no opportunity for inter-ascus mating. And so I'm wondering if inter-ascus mating could actually promote the rescue of nolosomic spores that failed to inherit a single chromosome. And so you might think this sounds crazy, but remember that one of the classic assays for chromosome loss in mitotic cells is this alike phaco or illegal mating assay, in which the loss of a chromosome 3 does not allow budding, but can be rescued by the act of mating with an appropriate partner strain. And you might say, well, haploid cells probably just can do this from some sort of residual mating, a maternal effect. But Jasper Reiner, the Hawthorne deletion, John Hawthorne had identified a mutation that could resulted from recombination between MAT and HMR and eliminated 59 genes, including 6 essential genes, and yet these spores could be rescued by mating upon germination. So I suspect that perhaps during meiosis between these interspecies diploids, a nolosomic spore could actually be rescued by mating with one of its sisters and could, especially in some cases, produce sort of a signature diploid cell monosomic for a single chromosome. And so finally, some of the experiments that I have ongoing right now are to recreate an interspecies diploid and look at the progeny of meiosis as they germinate. And you can see most of these spores are dead, but if I look in the streak of cells here, I can find many cases in which germination is occurring when I leave the asci intact. And so I want to then look at these and see if I can find this karyotypic signature of this inter-ascus mating rescue that I'm hypothesizing. And so this has many interesting, I think, questions more than answers. And I'm realizing I'm running out of time, and so I will just quickly skim over these and thank you for your attention.
Michael, what's the source of the estimate of 999 mitotic divisions per 1 meiosis?
I think those are mainly based on genomic analyses of sequence heterozygosities, basically, and how, as far as I understand it, and how frequently you would have to be undergoing meiosis in order to purify those to homozygosity. So I don't think there are very many direct observations of how often these things happen, because of course this is happening in the wild somewhere.
Hi. So you tempted us with two pieces of information. One, any idea what is leading to the genetic variation that you saw in the difference between bud versus mate choice? And second, what are the interspore bridges made of?
Yes, so we don't know what Ali saw, what could be underlying those differences. I mean, I'd love to take those strains and see if they do indeed have differences in the timing of the ascus wall breakdown. I think that would be a logical thing to look at, but we don't know what underlies those differences. And the interspore bridges, it's unclear exactly what they're made of. I think it probably might be chitin and other sort of things that you typically find in the septum of mitotically dividing cells, but it's still unclear, I think. Yeah,
Hi.
So,
Sorry, Mike. When people like us look at wild strains, it's very frequently in some of those strains that you see a tetrad that has a bud off of it, and sometimes at a frequency of 1 in 20 tetrads. Have you looked at that, and do you think that it's budding? Because one of the theories was that this was kind of an incompleted, and I never kind of bought that.
Oh, do you mean even prior to germination?
Yeah.
Yes.
Well, that was the theory I heard, but do you have a different theory?
No, I've seen that as well. In many cases, that just in the sporulation culture, there's lots of cases in which you find an intact tetrad with still sort of a remnant bud attached. And we don't know where those come from, and I'm not sure it's related to this at all, because I have a feeling those were formed before sporulation actually began, and yet for some reason cells in G2 were actually able to initiate sporulation. So I've seen it before, I don't have an answer for that.
Okay.
So do you think the cell wall, the ascus wall, has a signaling role, or is it a physical barrier?
That's a great question. I don't know what the ascus wall is a barrier to, and it's just a wall at this point, no membrane, so it's hard to imagine how much signaling could be going on there. But yeah, that's a great question. I don't really
Well, the carbohydrates certainly have plenty of signaling roles in other systems. So.
That's true, yeah.